nanomedicine-mediated drug delivery by advanced fluorescence microscopy techniques. Please.
Thank you for the kind introduction, and it's my pleasure being here. I thank the organizers for the invitation. So the title has been mentioned. I will talk about the use of advanced microscopy techniques that we have developed and also have been applying in the course of the years to study more precisely what happens with nanomedicine formulations in the different phases of the delivery process in the human body. So my smaller group is part of a larger group indeed, so I thought I would mention that as well. So it's called the Lab of Biochemistry and Physical Pharmacy indeed. So Jo de Meester, he's head of the lab, and this guy, I'm sure that many of you will know him, if not in person, then certainly in name. So he's in the nanomedicine field for many years, Stefan de Smet. He's also my previous supervisor, and now I'm happy to be colleagues with him. And he's heading the Ghent research group on nanomedicines. So honesty requires me to tell you that he's the one actually invited for this talk, but so he is now in the royal company in India. He had an obligation to go with one of the princesses of Belgium on a scientific mission to India, and he could not refuse. I'm sure you will understand that. So nevertheless, I will try to give a talk about what we do in the lab, and I will try to fill his place as well as I can. So I'm leading the biophotonic imaging group. We focus on biophotonic technologies to study the nanomedicine delivery process. So we are a very interdisciplinary research group. It's symbolized here. So part of what Stefan is saying, I'm not understanding, he does not understand part what I'm saying, but we try to have a maximal overlap to do novel science in these overlapping disciplines. So we are focusing on the delivery of biomacromolecules, mostly nucleic acids indeed, and we focus on non-viral gene therapy. We use mostly lipid systems or polymer-based systems. Polymers we do not develop ourselves. We collaborate with groups like Professor Henning or other groups who are focusing on polymer chemistry and development. In any case, as you know, these carriers, they are used to protect the macromolecules from degradation, to facilitate transport, and to have controlled drug release at the site of action. So if you talk about this delivery process in the body, after typically intravenous injection, I would say, then you have many biological barriers that these nanoparticles have to overcome. So you have to have stability in the blood circulation, extravasation to the extracellular matrix, mobility in the extracellular matrix, uptake into the target cells, and then endosomal escape and release from the therapeutics into the cytoplasm. So many different steps that need to be taken. And I guess it's clear for most of the people in the room, history has proven that this is a really challenging and very difficult task indeed. So we see in our group, and I think many of you, two challenges. On the one hand, we need better intelligent nanomaterials. On the other hand, to come to this, we need to better understand what happens to these particles. So, Stefan, so he's focusing on developing new material concepts, mostly delivery of nucleic acids, let's say, larger ones, smaller ones. And over the years, he has been using liposomes, biodegradable polymers, degradable nanogels. Now we're also using exosomes, very difficult, but all kinds of material concepts indeed, to have better delivery. And I focus on these microscopy techniques to better understand these delivery processes. So what happens to the particles in the blood circulation, what happens to them in the extracellular matrix, and what happens inside living cells. And we use, our experience is that these fluorescence microscopy techniques are quite useful indeed. So confocal microscopy, it's not special anymore, it's a standard laboratory technique nowadays. But there are more specialized techniques, more specialized tricks that you can play with these microscopy techniques that can give you more dynamic information on these systems. And in the course of this presentation, I will give a few examples. So if you think about administration through intravenous injection, the first questions already arrive. So we inject the nanoparticles in the blood circulation. The first question is, do these nanoparticles aggregate in the blood circulation, or do they perhaps dissociate? Does the cargo, the therapeutic cargo, perhaps, is it released from the carrier? And we would like to study this directly in the blood circulation system. And I will start with how we measure aggregation. because typically nanoformulations have a size of, let's say, 100 nanometer, nominal size. And considering the fact that the fenestrae in the blood circulation, in the capillaries, are typically between 100 and 400 nanometer in size, you can imagine that even the slightest aggregation of these 100 nanometer particles already alters the biodistribution indeed. So we would like to do sizing to measure aggregation of nanoparticles in the blood circulation. Now if you look at the available methods, you have many high quality techniques, great for laboratory purposes, but none of them actually suited for measurements in biological fluids. So we have electron microscopy, atomic force microscopy, great techniques, but you cannot apply it directly in the biological fluids. Light scattering, we use it all the time when we make formulations in the lab, but once you put it in serum, let's say, or in plasma, you have so many scattering components, you cannot tell anything from these signals. So then you have more specialized techniques like flow cytometry, Coulter counting, also great techniques, but they're not really suited for the nano range in our experience, and in any case you cannot fill these instruments with biological fluids. So looking at these problems, we realized a few years ago we need something else. We need a new method. And basically independently from that, so most of these things happen by coincidence, I'm sure. So coincidentally we were doing single particle tracking microscopy at that time. So what we do is we have fluorescently labeled nanomedicines. So mostly it's the nucleic acids that are labeled with a fluorescent dye. We put them in nanoparticles, and it means that thanks to the fluorescence you can see specific signals of these nanoparticles, of single nanoparticles, on this particular microscope in the biological fluid. And on this movie that you see happening, what you see is nanoparticles dispersed in plasma, and you see the Brownian motion. It's really random, but the thing is we can follow the motion on a particle by particle basis. We can do the tracking. I have to move on with the slide. So we do the calculation of the trajectories, and for each trajectory we can calculate the diffusion coefficient for each particle. And if you do this for 1000 particles, you get the distribution of diffusion coefficients, and if you know the viscosity, then you can convert this to a size distribution. It's basically the same as a DLS, a dynamic light scattering instrument that also measures the diffusion and then converts it to a size distribution. So I will not talk about, this is what the microscope looks like. Basically, it looks a little bit complicated, but basically it's a fluorescence microscope. We use laser illumination because it's a little bit more efficient, and we use a very sensitive CCD camera. Now, so I'm sure that you understand the concept of the sizing behind this technique. So we track, we measure diffusion, and we convert it to a size. To show that it works in biological fluids, we did this experiment. So we took fluorescent nanoparticles, nominal size 0.1 micrometer. We measured them in buffer. The actual size was about 140. We also dispersed them in serum and in whole blood. We do the tracking, and as you can see, by taking into account the viscosity of the solutions, we can have these very accurate size distributions and average sizes. So we use this system now to study, for example, the aggregation of cationic liposomes that are being used for nucleic acid delivery. We compared non-pegylated ones, pegylated ones, and we started off with this very simple artificial blood circulation system. So it makes use of plastic tubing with a diameter that is comparable to the lateral tail vein of a rat. We took human blood in this case, so we have many students that had to donate a little bit, you know how it goes. So this is student blood. So what we do is we have a vessel with this blood, and then we have tubing going out, coming in, a very simple pumping system. Everything is put at 37 degrees, and this is our artificial system. At time 0, we inject a bolus of these fluorescently labeled liposomes, and as a function of time, we take samples from the blood compartment. And this, you can imagine that this kind of resembles the heart, let's say, the central blood compartment. So the advantage of this system is that, of course, we do not need an animal, of course. And then secondly, suppose that aggregation happens in an animal. I will show you that this is the case. Aggregates are rapidly cleared, but we want to measure aggregation. So in this system, everything that aggregates remains in the system, so we will be able to measure it. The first thing we did is compare different biological fluids to appreciate the difference in aggregation profiles. So we injected non-pegylated liposomes in human serum, plasma, and blood from the same person. And what you're looking at here are those relative size distributions. So this is the relative fraction of different size of liposomes measured in serum, plasma and blood as a function of time. So the colors mean different time points. We inject it and we take samples at different time points. In serum you see, as a function of time, slight aggregation, not unexpectedly, but in plasma you see it's already much worse, and if you go to the whole human blood, it's an order of magnitude worse. So in our experience from this experiment, we know if you want to appreciate the full aggregation probability of your particle, try to do it in undiluted, true biological fluid, not derivatives, which are easier to work with. Of course, what this means is if your particle already aggregates in serum, for sure it will aggregate in blood. So let's move on. We compared with pegylated lipids, with pegylated liposomes indeed. And we were wondering how much of the PEG needs to be included in order to avoid this massive aggregation in blood. So this is the situation without PEG, massive aggregation. 1% of pegylated lipids, no change. 3%, no change. 5%, here something happens. You see, there's still aggregation, but it's already much less, as you can appreciate. But only from 10%, we could keep these liposomes stable in blood for at least 2 hours in this system. So 10% is the least amount, according to our finding, that should be included. And then we went on to an in vivo experiment where we injected these pegylated and non-pegylated liposomes to see what happens in vivo. We inject the rat. We take at different time points small blood samples, so the rat is not euthanized, so we just sting it a little bit. That's the only thing that the rat was suffering in this case. Very small samples. We bring it to the microscope and then we do these particle measurements. And I will show you two extremes, so that you appreciate visually what happens and what is the difference. So pegylated liposomes, you see they move very rapidly. They remain quite small. These non-pegylated ones, they aggregate, and you can see this already on site that this is a very big aggregate indeed. So we did this more carefully. This is what happens with a non-pegylated liposome. So relative size distributions again, the rat was injected and we take a sample at 5 minutes, 20 minutes and 40 minutes. At 5 minutes, we see massive aggregation, as we expect from the artificial system. And then interestingly, as a function of time, the aggregates were actually removed from the blood circulation. So we know from this experiment, if you have aggregation, it's quickly removed from the bloodstream. Non-pegylated, the pegylated ones, indeed, they remained stable, no aggregation, also not at initial time points, and it keeps stable for a long time period. Longer time period. So PEG decreases aggregation, as one would expect, but we can measure indeed, and it improves the circulation time. I will come back to that. And to show you that this is not limited to liposomes, this was a study on cationic dextran nanogels in which siRNA was included. Also here, so this was in plasma, also here you see as a function of the pegylation degree that you keep the particle more stable if you include more PEG. Now, we can do more than just size measurements. We can also have very accurate number concentration measurements, which is not possible by DLS, for example. And okay, so the physical details, this was together with a statistics group. It's a very nice method, but I will not go into the mathematical details. You can appreciate on site, or you can imagine that indeed by counting the trajectories that you have information on the number concentration of particles. More trajectories means more particles. This precise relationship, it's a bit complicated, but there's a mathematical model to relate those two. And these are the proof of principle measurements. So we have dilution series and absolute number concentration. So this is 10 to the 8, that is on the top here. So 5 times 10 to the 8 particles per ml. And the solid lines are the expected concentration values. The dashed lines are the uncertainty provided by the manufacturer. So within this uncertainty, we can indeed have accurate absolute number concentration measurements with excellent accuracy and precision. So we apply this to the data that I showed you before. The in vivo experiments with non-pegylated and pegylated liposomes. And here we can have a quantification of the number of liposomes as a function of time remaining in the blood circulation. So it's not just based on an overall fluorescence measurement, it's not just an overall radioactivity measurement, it's really on a particle by particle basis that we know the concentration. And now you appreciate that the pegylated particles, the circulation, they are also being removed from the circulation, but in absolute numbers they remain much more there than the non-pegylated ones, which aggregate and which form fewer particles indeed. So we can follow indeed the clearance kinetics on a particle by particle basis in blood circulation. Then, let's talk about dissociation. That was one of the questions at the end of the last presentation. So one question is, suppose that your particle does not aggregate, that's great already, but maybe the cargo is released in the blood circulation by competition with serum proteins, for example. The way that we measure this is by another fluorescence-based technique. It's called fluorescence correlation spectroscopy. It seems like a complicated optical drawing, but basically this is just a confocal microscope, and it's actually a simple confocal microscope. In a confocal microscope you have the focused laser beam that is being scanned to make an image. In FCS, fluorescence correlation spectroscopy, it's just a stationary laser beam. The beam is just standing there. And this is a magnified illustration of this focused laser beam. And what we do is we just wait over there for molecules to arrive. And when a molecule arrives in the beam, you get an increase in fluorescence. And when a molecule leaves, you get a decrease in fluorescence. So you get these seemingly noisy profiles. So this is the fluorescence intensity as a function of time. But every fluctuation is actually a molecule coming in or moving out. Now you can imagine that if a molecule moves fast, that you have very fast, thin fluctuations. If you have a slowly moving molecule, you will have broad and slow fluctuating profiles. So the right mathematical analysis, what you can derive from such experiments is the concentration of fluorophores, but also the diffusion mobility. You can do it in two colors as well, but I will not talk about that. Look at this. So this is an illustration of what happens. So we would like to use now FCS for measuring the complexation efficiency of nucleic acids inside nanoparticles. So we start off with the fluorescently labeled nucleic acids, siRNA in this case, in buffer solution. And if you do an FCS measurement, this is what the experiment looks like. So the intensity, the average intensity here, is proportional to the concentration of molecules, of course, the average number of molecules in the detection volume. And these small fluctuations are happening because of molecules moving in and out. The point is, if we now add the nanocarriers to form complexes, we will form polyplexes, lipoplexes. What happens is this. siRNA molecules are captured in the nanoparticles, so the overall concentration of siRNA molecules decreases, which means that the baseline here is dropping. We have less free siRNA, but on top of this baseline, now we have these peaks emerging, bursts of fluorescence, coming from siRNA complexed into nanoparticles. So this is a confirmation indeed that complexation is happening. And the point is that the difference between the original level of free siRNA fluorescence and the baseline that is remaining after adding the liposomes, this difference is proportional to the siRNA molecules that are actually included into the liposomes. This is why the baseline is dropping. So we can measure complexation efficiency, and why we're doing this is because in the end we want to measure the inverse, we want to measure the dissociation. So we start from this, we put it in serum, and then we see if the baseline increases again because siRNA is released. Validation experiment. So typically, how would you measure complexation of siRNA into a nanocarrier? Typically we do this by gel electrophoresis. So what we did is we complexed siRNA with pegylated liposomes on a 1:1 charge ratio, a 5:1 charge ratio, and a 10:1 charge ratio, so increasing number of liposomes. 1:1 shows you that it's insufficient complexation, but starting from 5 it should be okay. And the same was measured with this FCS technique. So this is the 1:1 result. And indeed, so the gray signal is the free siRNA. And if you add this little amount of liposomes, indeed you see only a very tiny amount of decrease of the background line, because only very little siRNA is complexed, and very few peaks indeed. But if you include more liposomes, then indeed you see a bigger drop of this baseline, because more siRNA is included, and you see more peaks happening. So this nicely corresponds to what we see in a gel electrophoresis experiment. And now we use this method to compare two formulations of lipoplexes, two different preparation methods. This is what we call the classic method. I think this is what is usually being done. So you have a lipid film, you hydrate it with a buffer solution, you form the liposomes, and then this is added to siRNA. And our hypothesis is that in such a system, you are complexing siRNA, but where does it go? Probably it's just sitting on the outside. Yeah? It's attached, but not really protected. As an alternative system, you can do this hydration directly with the solution of siRNA. With the solution of siRNA. And then if the liposomes are formed, statistically, what will happen? Well, 50% will be inside and 50% will be outside. So if we put those two formulations in serum, we would expect a difference in potential release of the siRNA by competition with the serum proteins. So we did these measurements. So those are the two formulations. We incubate with 90% serum. We measure by FCS, as I just have explained. And indeed, compatible with our hypothesis, in this case, we saw 90% release of the siRNA in serum, because this is actually just sitting outside, while in this case we saw 50% release. 50% is still a lot, but at least you keep something inside. And measurements like these tell you something, that this formulation, for example, will never work in vivo. You don't have to go in vivo. Just do the in vitro experiment and you will know beforehand that it will not work. So I think these experiments have value to reduce in vivo experiments. It's not limited to liposomes indeed, so this was a polyplex combination of siRNA with polyplexes. Also here it could be measured that there is a good correlation between the dissociation in serum and the lack of transfection efficiency indeed. So intravenous delivery is one thing. It depends on the pathology, of course, whether you would go intravenously or through another route. For example, another application that we have in the group is the treatment of biofilm infections in the lungs of cystic fibrosis patients. So you know, in cystic fibrosis we have this, or patients have this very sticky, thick mucus in the lungs, and bacteria can easily stay there for a long time, and they colonize this mucus and they form bacterial biofilms. The bacterial biofilm, it's indicated here, it's a very dense cluster of bacteria, and they also secrete an exopolysaccharide matrix, a slimy matrix that protects them from the environment. So the combination of this exopolysaccharide matrix and the dense cluster that they are forming makes them very hard to be killed by classic antibiotic therapy. So what we would like to do is to develop a nanomedicine formulation that can penetrate through the sticky mucus, that can penetrate into these dense clusters and give them a sudden release of a high doses of antibiotics. Now the first thing that you have to wonder about is what kind of nanoparticles have this type of properties? How should we make a nanoparticle move through these very dense and sticky biolayers? And we studied this by model nanoparticles, polystyrene, fluorescent particles. We have them carboxylated, so a negative charge, aminylated with a positive charge, and pegylated to shield the charge. And these are in vitro biofilm experiments. I will start the movies. I hope you can see it. The contrast is not so great. If you're interested, I can show you the movies on the computer afterwards. You're looking at single particle tracking movies in two colors, but it's just grayscale, unfortunately. So here on the left side we have the green image coming from the nanoparticles. On the right side we have the biofilm bacteria labeled with the red dye. So, but you cannot really see them very well. So here are some bacteria. Here's a cluster of bacteria, as you can see, a very dense cluster. Also here you see it a little bit better. These are nanoparticles, positive charge. They just get stuck to strands of DNA in the biofilm because of lysed bacteria, the DNA. The negatively charged, they get stuck to the bacteria themselves. And the pegylated ones, I don't know if you can see it, yeah, they move quickly. Yeah. Can you see it a little bit? This might be a little bit more clear. So I'm showing here the same situation. The fluorescence is the biofilm bacteria, and on top of that I've plotted these mobility trajectories. So this is pegylation, negative charge, positive charge. And you appreciate immediately the difference in mobility of the pegylated nanoparticles. You see free, brown in motion, while the charged ones, they are basically stuck to all kinds of components. And if you calculate the average mobility, so relative to water, the pegylated particles, even in the dense clusters of the bacteria, are actually almost as mobile, while the negative and the positive charged ones, they have, they bind and they have very low mobility indeed. And we can have these continuous distributions of diffusion coefficients, so we can quantify this quite accurately. This is the situation in mucus, because this is also a barrier that needs to be overcome. So we take a really clinical sample, so sputum from cystic fibrosis patients. Yes, so indeed, pegylated particles, negative, positive, and again you appreciate, I think, the difference in mobility. That is obvious. So after quantification, we see that clearly. So these are the pegylated, the first four. Pegylation clearly improves mobility. And it also shows that actually it's not the biofilm that is the hardest barrier, it's more the sputum, the sticky mucus, that is more difficult to penetrate, lower mobility. And charges should be avoided. That's what we learned from this. How am I doing all the time?
10 minutes.
All right. This is another application that we're working on. So it's retinal gene therapy. So there are a number of retinal dystrophies that have a genetic cause. So bringing nucleic acids to the retina, and then especially the photoreceptor cells, or this final layer, it's called the retinal epithelium cell layer. These are phagocytic cells that are there for the homeostasis of the retina. But those two cells are related to genetic diseases of the eye indeed. And we would like to have genetic genes delivered to there. And topical administration is not an option because you have too many layers to cross, too many barriers. Going through the bloodstream is also not an option because you have a blood-retina barrier, similar to the blood-brain barrier. So either it's called subretinal injection, it's surgery where they open the eye, they take out the vitreous and they inject it underneath the retina is one option, but of course you will do this only once in your life, it's not suitable for repeated administration. The other is still not very pleasant, I think, but it's an option. It's intravitreal injection. So the patient has to go to the hospital, and it's a trained nurse that can do this. So they open the eyelids with a metal thingy, I don't know the name in English, and the patient has to look to one side, and then they quickly inject with the needle into the white of the eye, into the vitreous. So the vitreous is what is inside the eye. So this is a cow eye. If you open it, this is what is inside. It's actually a hydrogel. It's really a physical gel. You can take it out, you can put it on the table. So it's a really physical hydrogel. It's made up of collagen fibrils, and there's also lots of hyaluronic acid inside. And of course lots of water and salts and ions and so on. So when we inject it in the vitreous, it comes in this jelly material. So again, we are wondering about mobility properties here, and we took those same particles to study the mobility. And originally, so this is almost 10 years ago, we studied this by FRAP. It's fluorescence recovery after photobleaching, one of these other interesting fluorescence techniques. The concept is very simple. You have a fluorescent sample, these beads for example. You can photo-bleach them with a high-intensity laser beam. You destroy the fluorescence. And the photo-bleached molecules or particles, they will move out of this bleached area, and this area is replaced by intact particles or molecules. So as a function of time, you get a recovery of the fluorescence inside this bleached spot. So this would be an actual experiment. Fluorescent sample, you bleach a part, and as a function of time, you see a recovery because of diffusional mobility stability. And this can be quantified indeed to have diffusion coefficients. So we did a lot of theoretical development on this in the course of the year. So the first experiments that we did is we actually took out the vitreous gel and then we physically mix it with these particles. We do FRAP experiments indeed on the vitreous gel here. Yeah, you can do these mobility measurements, and then we noticed that relative to water, larger particles have more hindrance to move through it as compared to smaller ones. And this could be caused by steric hindrance. So not just viscosity, but by steric hindrance because of the polymer network inside the vitreous. However, yeah, when you look at these vitreous images on the microscope, you see that particles are mostly getting stuck in these linear structures. Those are the collagen fibrils. But we were a little bit doubtful about this model because, you know, when you take the vitreous out and you put it on the table, immediately the water starts leaking out. And then we start mixing it, so you can imagine that the structure after these manipulations is maybe not the one that's actually inside the eye. So we wanted to have a better ex vivo model, and this is what we did in recent years. So Thomas Martens is the guy who developed this. So what we do is we go to the slaughterhouse, we have an eye of a cow, and what we do is we just take out the lens, so just the front part is taken out, and then you have a window into the vitreous without actually damaging the vitreous. And the vitreous does not leak out because the vitreous is enclosed by a limiting membrane, so it's actually protected. But you can image through this membrane. So then we put this eye without the lens upside down in a petri dish with a glass bottom indeed, so that we can do microscopy. And then we inject the particles near the observation volume. And then we get these particle images. So positively charged beads. So this is the raw movie and this is the processed movie with the trajectories. These are anionic beads. Yeah, looks not that much different, a little bit more mobile perhaps. And then pegylation, no surprise, massive mobility. And this could be quantified indeed, so the PEG mobility is very high. Negatively charged particles have a little bit lower mobility, and then positively charged particles, but this is actually what is mostly being used, cationic particles for gene therapy. They have an interesting bimodal behavior, so lots of them get stuck. They get stuck to the collagen fibrils, we found out. And then another part, and this is quite interesting, they are nevertheless mobile. And we figured out that this is very likely due to coating with the negatively charged hyaluronic acid molecules. So when the particles are injected, if they get coated by the negatively charged hyaluronic acid molecules, before they bind to a collagen fibril, they have a protective coating that keeps them mobile. And this is a strategy that we are currently pursuing to replace PEG by hyaluronic acid coating indeed. This was also applied to actual carriers, not just model particles. So this was in collaboration with Professor Engbergsen. I'm sorry, many people will know him, I'm sure. He's also a polymer chemist, and he developed these bioreducible polymers. It's cysteamine bisacrylamide aminobutanol. So ABLE is this side chain. And they are very good in efficiently complexing nucleic acids. And due to this disulfide bond, it's also bioreducible in the cytoplasm of the cell. Any case, so those particles were injected. And again, we see that, so this is mobility in water. If you inject them into the vitreous, the cationic particles, it's a blue line. Again, this bimodal behavior, it's similar to the artificial particles. And then if we use a pegylated variant, indeed, relative to the blue curve, you see that the mobility again increases. So this is very nice. But as we all know, pegylation is nice for mobility and stability, but it inhibits cell uptake because you also prevent attachment to the cells. And this is where hyaluronic acid might be a better alternative, indeed. So, I discussed a little bit about stability in the blood circulation. Other extracellular matrices were discussed, so vitreous and biofilms, mucus. So what about what happens in the cell? Do I still have time? Or? Yeah, okay. So if you look at what happens to the cell, so complexes will arrive at the cell membrane, they will attach to proteins or polysaccharides indeed. They get stuck at the membrane, they are typically endocytosed, they are transported through various organelles in the cell. At one point, we don't know where exactly because it's difficult to study, we have endosomal escape. After endosomal escape, there's dissociation of the nucleic acid from the carrier, and then in case of DNA, it should translocate to the nucleus. So these are the steps that needs to be investigated to understand why some formulations are better than the others. So the first thing that we studied is the cellular uptake, so endocytosis. And I'm sure that many of you will know that there are many endocytic routes into the cell. Endocytosis is not just one phenomenon. There are many types of endocytic pathways into the cell. And we would like to know what is the successful pathway of these many pathways, which ones are linked to most optimal transfection efficiency for different types of particles. And this can be studied by a use of a combination of pharmacological inhibitors that inhibit certain pathways in combination with the knockdown of specific endocytosis-related proteins. And this is a study that we did with an endocytosis expert. I'm not an endocytosis expert, but we did the study indeed together with him. It's Dr. Arwin Jones. And so we studied through which of these many pathways that are currently known. So we have caveolin-mediated endocytosis, clathrin-dependent endocytosis, flotillin R6, click-G pathway, macropinocytosis, phagocytosis, and many more pathways are being discovered basically every year. So, but those are the ones that were known when we did the study. And the thing is, so by using these pharmacological inhibitors or knockdown of proteins, we shut down one by one each of these pathways and evaluate the effect on particle uptake and transfection to see which one is the most important in the uptake of these polyplexes. So Dries Verkouteren is the PhD student who did these studies. So the bottom line here, I will not show the data. Why? Because it took us 3 years to figure this out. This was a very much underestimated study. And I will never do it again because we figured it out, but it's really not worth the effort, in my opinion, at this stage. Why? Because these pharmacological inhibitors and even the knockdown, they're not so specific as people would like them to be. So you downregulate perhaps one pathway, but other pathways are upregulated, or there's a combination of pathways that are being downregulated, and you get very convoluted results, and you have to do many repeats and control experiments to kind of figure out what's happening. I think in the end, after 3 years of hard work, thanks to this guy, I would have stopped it, but he persisted, and I think he figured out what happens. So out of these many pathways, and out of many data and graphs, he figured out that it's actually a relatively newly discovered endocytic pathway that is involved in these polyplex uptake. It's the flotillin 2 dependent pathway on the one hand, and on the other hand he found out it's also phagocytosis. Not so surprising because the cells that he was using are those retinal epithelium cells, the cells at the back of the retina of the eye, and those are phagocytic cells. So it's not so surprising that this was involved as well. Then he also figured out that of those two pathways, it's actually only the flotillin 2 dependent endocytic pathway that contributes to transfection. Particles that are taken up by phagocytosis in those cells, I don't know how this translates to other cells, phagocytosed particles did not contribute to transfection. Those particles were just trafficked to the endolysosomes for degradation. And it's only those uptake, those endocytosis particles, endocytosed particles that contribute to the transfection efficiency. Yes, more recently we also were interested, so we know how they are taken up, but what happens in between? How are they trafficked inside the cell? You have many different types of endosomes. Which ones are involved? So a particle enters, maybe it's sorting endosomes that are involved, late endosomes, endolysosomes, maybe also these autophagosomes might also be involved. So let's figure it out. What we did is we transfected cells with GFP fused to proteins specific for these different types of endosomes. For example, these are Rep5 labeled endosomes in green. Rep5 is a protein associated with early and sorting endosomes. And in red, what you see are the fluorescently labeled nanoparticles, the polyplexes of which the DNA is labeled. And you can appreciate that, for example, at this time point in the cell, you see that the two are moving independent of one another. So there's no association between those two structures at this time point. But the Flotlin2 endosomes, which were involved in the uptake, if you label them on the microscope, indeed you see there's a nice correlated movement of the nanoparticles with these structures, so they're really associated and being transported in the cell by these kind of vesicles. So this confirms also the uptake experiments. We analyze this quite meticulously. We have an algorithm developed that looks for correlated motion. It's a better measurement for colocalization than just looking if the color is a little bit yellow. You know, it's a little bit more tricky. But if you have correlated motion, you're for sure that you can be certain that the two are related. So so just to show you the principle, so we have these two color movies. Red is a polyplex, green is an endosome. They are split in the respective channels. We do the tracking, as always. And then we look for correlated trajectories, correlated motion indeed. Yeah, this is done by software. So the blue indicated parts are correlated trajectories. Long story short, he really took over 1,000 movies. Can you believe it? He was a really great student, Dries Verkouter. He took more than 1,000 movies of these particles added to RPE cells, labeled with different types of endosomes, of course sequentially. Not all these endosomes were labeled at the same time. So one time he studied the Flotlin2 endosomes, and then later on the late endosomes, and so on. In the end, he got this very nice, very detailed, I think this was the first really detailed study of intercellular trafficking to my knowledge. So as a function of time in the cell, this is what happens. So this is percentage of colocalization with different types of endosomes. These are the colors as a function of time. And you see, for example, not so surprisingly, but this is quantified, that at early time points, so after 20, 30 minutes, we have high colocalization with Rep5 endosomes, these early and sorting endosomes. But then it quickly drops, and where are the particles going? Well, you can see it. They are transferred to these late endosomes, and a little bit later on, they're going to the endolysosomes. And this is the major problem on the intercellular level. So most of the gene complexes, they just end up in endolysosomes. And we need to come up with strategies that avoid this degradative pathway. We should get them out in some way or another before they reach the endolysosomes, where they are mostly lost probably. So we figured out that we have uptake through phagocytosis and Flotlin2 mediated endocytosis. We have trafficking through early endosomes, late endosomes, endolysosomes. We don't know yet where endosomal escape happens. Unfortunately, we didn't find it, which again signifies that this is a relatively rare event. But we are still looking for it. And interestingly, we also found this contribution by the autophagocytic pathway. So we assume that even when a particle escapes from the endosome, it might again be enclosed by autophagosomes and still be transported for degradation to the endolysosomes as a protection by the cells against foreign particles. So, in summary, I hope to have given you some examples of where advanced microscopy techniques are helpful to understand a little bit better what happens to nanomedicines in different phases of the delivery process in the body, starting from the blood and other extracellular matrices up until what happens inside living cells. So I'm from Ghent. This is the typical view from Ghent city. We have these three towers here, 1, 2, 3. They're actually on one straight line. A very nice historic town. I advise you to visit it for one day. It's very nice indeed, by night. So I thank many people. So of course this was not all my work. I presented what happens in the group. I thank Stefan, I thank Jo for their guidance. And if you have an interest, we always are open to collaborations. If you think I would like to try this assay out on my particular type of formulation, we can always talk about it, how to proceed with that. So thank you very much for your kind attention.
So thank you very